The pH or environment is really important for a protein's structure and function, and we'll see how buffers can help balance out the pH. And remember that pH does define the concentration of protons that are available in the system. By the end of this lecture, you should be able to calculate the pH of a buffer using the Henderson-Hasselbalch equation. We'll also look at how the pH can influence the ionization state of amino acids, so whether they are protonated or deprotonated, and if you know what the pH is, you should be able to predict the ionization state of the functional groups. You'll also have an idea of how to pick out a buffer in order to study a protein, and whenever you go forward and read a journal article about a protein, you'll see that the experiments are conducted using a variety of different buffers. So after this lecture, you should be able to understand the importance of these buffering systems in order to maintain pH. These are the supplemental readings for this lecture, just a review of the 20 amino acids as well as some acid-base chemistry in case you forget it from your chemistry prereqs. And we'll also look at the end of the lecture of a specific example where pH has an effect on oxygen transport, and this is known as the Bohr effect. We've been discussing the structures of amino acids, and up until this point, we've been drawing them with a positively charged amine, or NH3+, and a negatively charged carboxylic acid group, or COO-. Recall some of the amino acid side chains also have positive or negative charges on them. But how do we know if these functional groups are protonated or deprotonated and adopt these charges? In order to understand this, we need to review the importance of pH and how the environment contributes to the ionization of these functional groups. So remember that protons, or H pluses, as well as hydroxide ions, can be formed upon the dissociation of water as well as other weak acids that can donate their protons, or bases that can be proton acceptors. And remember the relationship that pH is equal to the negative logarithmic value of the concentration of protons. So if there are lots of protons or a large concentration of H+, you will have a lower pH. So at lower pH values, we have more protons that can actually protonate or join with functional groups in order to form groups such as that NH3+. So the pH of the solution is really important since these functional groups that we see on amino acids can also act as weak acids or bases. They can either lose their protons or accept them depending on what the pH of the environment is. There's also another constant value called the pKa value, and what the pKa value tells you is the strength of an acid. On the right is just a scale showing you the various strengths of acidity of different fluids. And remember, at high pH values, we have low concentrations of protons. The pKa value is obtained from the negative logarithm of the acid dissociation constant, or Ka, where Ka measures how much of the acid, or Ha, dissociates into protons and the conjugate base, or A-. Strong acids will have high dissociation value, Ka values, and low pKa values. pKa values are useful because they tell you the pH at which a functional group loses or gains its proton. If we look at some of the functional groups that we've seen before, they all have very different pKa values. Now, you don't need to memorize these. These would be given to you on a quiz, but you do need to understand that the pKa value is the pH at which the functional group is protonated or deprotonated. Starting at the top for the carboxylic acids, the pKa is lower, and this is the pH at which we go from the protonated form on the left to the deprotonated form, or the conjugate base, on the right. So at a pKa value of 3.75, formic acid would be accepting and donating protons. Some functional groups like phosphates or phosphoric acid have multiple protons that can be lost. So at very low pH, we have lots of protons present, and we have the completely protonated form shown on the left. And as we increase up the pH scale, we start to lose the concentration of protons one by one, and at different pKa values or pH values, we will start to lose different protons. So pKa 1 will start to lose the first proton, pKa 2, pH 7.2 will start to lose the second, and then once we're up in the pH range around 12.4, we're going to start to lose that third proton. Phosphate buffers are great because around the pKa2 value of 7.2, this is close to physiological pH, so we could have a mixture of these two forms of the molecules that will act as buffering species. The weak acid on the left and the conjugate base on the right of that pK2 arrow. At very high pH values, this is where the amines also start to be deprotonated. So this is why at physiological pH, we've been drawing the amino groups as protonated NH3 pluses, since the pH is below the pKa value and it's going to be in the protonated form. Now the Henderson-Hasselbalch equation can be used to determine the pH of your solution depending on how much weak acid we have present, so the amount of protonated HA versus the amount of deprotonated conjugated base. And this is all derived from the acid dissociation constant equation for Ka. Okay, you don't need to know the derivation, but definitely should know how to use this equation, sort of these plug and chug type questions, where we can actually figure out what the solution pH is based on the concentration of these species that are present and the pKa value for that functional group. Okay, so if you have a mixture of a weak acid and a conjugate base, you'll see that we can figure out what the actual pH value is. Okay, so you don't need to know the derivation of the equation, just how to use it. So this relationship between pKa values and pH is that when the pH of the solution is equal to the pKa value, you'll actually see a 50-50 mixture of the protonated form of the functional group as well as the deprotonated form of the functional group. Okay, so if you were to plug in concentrations to that Henderson-Hasselbalch equation that I just showed you, you would see that the pH would equal the pKa when you have equal amounts of A- and HA. Remember, proteins have lots of different functional groups that can be protonated or deprotonated, and really which state they're in will allow for them to form those non-covalent interactions. What a buffer can do is it can act as a solution of weak acid and conjugate bases that will resist any major changes in pH. So if you were to stick a bunch of protons present into the solution, the buffers are going to bind to those protons instead of the protein's functional groups, and this is what can help maintain a protein structure and or its function. Okay, so the buffers really are neutralizing these small additions or losses of protons that will keep the pH stable so that we can still maintain these interactions between the amino acid side chains. There are lots of different buffering systems, but when you're looking at which buffer to use, generally you're looking for a buffer that will maintain the pH around plus or minus 1 pH unit around the pKa value. And this is something you can actually do if you're going to take any of our upper year lab courses, such as BCH 370. You'll actually look at different buffers and be able to elucidate which ones you would use in an experiment. So what you're going to do is select a buffer that has a pKa value closest to the pH that you want to maintain for your protein. And this also changes depending on which temperature is present. Okay. The other things to sort of think about are the chemical stability of the different buffers. There could be side reactions that will occur and produce other molecules that could affect your experiment. So really the interference with your experiments is one thing to consider, as well as the cost or the availability of producing these types of reagents. So if you are looking for lab experience, once you complete BCH 210, 370 is another course you could take. We usually talk about physiological pH being 7.4, but keep in mind that proteins can exist in different environments that have different pH values. More acidic environments like the stomach, where molecules are digested and broken down, as well as the lysosome within a cell, may have higher concentrations of protons that can lead to the protonation of the various functional groups. You don't need to memorize these numbers, but do appreciate that when it comes to purifying and studying a protein, the pH is a very important consideration. In the PDB protein structure assignment, you will look at a specific protein and how it was purified and characterized, and what buffers were used to maintain a constant pH. And sometimes this isn't a physiologically relevant buffer for various reasons. Up until this point, we've been drawing the amino acids with a positively charged amino group and a negatively charged carboxylic acid group. You should now understand why these groups have the charges that they do and why we consider some amino acids to be Zwitter ions if they have a neutral side chain, with the positively charged amino group and the negatively charged carboxylic acid group canceling each other out. So at physiological pH of 7.4, we are above the pKa of the carboxyl groups, which vary slightly, around 2 depending on the environment, so it will be deprotonated or a COO minus group. And below the pKa value of the amino group, around 9 or 10, so this will result in the nitrogen being protonated to give us the NH3 plus groups. Some of the side chains can be protonated or deprotonated, so you should be able to recognize the negatively charged amino acids on this table and that have side chains that are deprotonated at physiological pH. Now we do use the terms sort of interchangeably, so aspartate and aspartic acid, where aspartate represents the conjugate base and aspartic acid represents the protonated form. These terms are used interchangeably, but at physiological pH, it would be aspartate that is present. There are also the positively charged amino acids, lysine and arginine, that are protonated and positively charged at physiological pH. While histidine is a special amino acid in that its R group has a pKa of 6.0, so close to physiological pH, so it is frequently protonated and deprotonated depending on its function. There are also some hydroxyls and the thiol that can lose their protons depending on the chemical environment, and these are also listed in this table. You may remember titration curves from your chemistry course, or may have even done a titration yourself. And this is where we take a species of molecules and either deprotonate it or protonate it and watch how the pH changes. So in this case, we're going to look at the titration of histidine, starting at a very low pH, and we're going to start to add base in order to neutralize protons. And if we measure the pH on the y-axis, we can actually look and see how the pH changes as we are deprotonating the groups. So if we start with the completely protonated form of histidine, shown on the left, that will have an overall charge of +2, we'll start to see that the first functional group that loses a proton, i.e. the one that has the lowest pKa value, will start to lose its proton as we add base in. So remember, the pKa value is the pH when a group starts to be protonated or deprotonated. So in this region, the carboxylic acid group will start to lose its proton, become negatively charged, and we'll start to see a slight increase in pH. We call this region of the curve the buffering region, and this is where we have lots of molecules that are buffering or binding to the protons. And that's what's going to resist a huge change in pH. So histidine will be giving up its proton from the carboxylic acid group, neutralizing the OH that's being added and producing our conjugate base. The pH will slowly rise and we're not going to see a huge increase in pH on the graph. Remember, the buffering region is usually within 1 pH unit around the pKa value. So this is the flat portion of the graph. Eventually, you will have completely deprotonated all the carboxylic acid groups, and the molecule that was present will have a +1 net charge. You'll start to see a loss of another proton at a pH of 6.0. So this is the pKa value for the imidazole ring, in order to reach the Zwitter ionic form of histidine. Now, in order to calculate the isoelectric point, so this is the pH when the molecule has a net charge of 0, we actually average the two pKa values on either side of this zwitterionic structure. Okay, so we would average the pKa values of 6 and 9.2. At a pH of 7.6, the majority of the species present will be this zwitterionic form. So this sharper increase in the curve where we're between the two buffering regions. At this point, the imidazole side chain is not protonated, and the positive and negative charges on the amino and carboxyl groups will cancel each other out. So this is known as the isoelectric species, or the isoelectric point, or PI, where it's the pH where we've averaged the two pKa values around this species in order to determine at which pH value do the majority of the species be present in the zwitterionic neutral form. As you start to add more hydroxyl ions, we start to lose the protons on the amino group, and this is the third buffering region to eventually end up with the deprotonated histidine with a charge of -1. You could then titrate your way back down the curve, adding more protons, where you're going to need 3 equivalent concentrations of protons to protonate the 3 functional groups. So just to summarize, proteins have many functional groups that are important for their structure, either through the formation of covalent or non-covalent interactions. And this can be influenced by the pH of the environment. The pH can also affect hydrogen bonding capabilities that might be important for enzyme function or the ability to interact with water and other binding partners. The pKa value, remember, is the pH at which a functional group will be protonated or deprotonated, and this does vary depending on the chemical environment of that functional group. So there's not always one specific value, as we can see for the amino groups on all the different amino acids. And it's where buffers come in handy because they can maintain a stable pH in order to provide those weak acid and conjugate base species present that will bind or accept or lose protons. Okay, and these will be very helpful when we're trying to study a protein or its biological function. Your blood has a very important buffering molecule called bicarbonate, or HCO3-, which has the ability of maintaining the pH around 7.4. This graph shows the percentage of buffering molecules present at different pH values. So at physiological pH, roughly 7.4, there are lots of bicarbonate molecules that can either accept or donate protons. If the concentration of protons increases, bicarbonate binds to them and produces carbonic acid, or H2CO3, which can break down to the waste products water and carbon dioxide. So whenever you're exhaling CO2, it might be from metabolized chemical reactions or due to the removal of excess protons. This prevents those protons from binding to important biomolecules and affecting their structure and function. One example of how protons can influence a protein's function is the Bohr effect, which is important for the release of oxygen in the tissues. So in the tissues where metabolism is occurring and producing lots of CO2 as well as protons, a lower pH can actually result in the protonation of an important histidine residue in hemoglobin. This histidine, so histidine 146, can actually form an ionic interaction or salt bridge with aspartate 94, and this is shown in the bottom right. This will change the overall structure of hemoglobin so that it can release oxygen. The graph on the top right just shows how in different pH environments the percent saturation of oxygen of hemoglobin changes, where at lower pHs there can be less oxygen bound. You can see that in the tissues where there is less oxygen or a lower partial pressure of oxygen, without a lower pH there would be more oxygen bound to hemoglobin, or 34% bound saturation in the pink curve versus 23% bound at a pH of 7.2 in the blue curve. This is one example of how the pH can influence a protein's function. The take-home messages for this lecture are that the pH of a solution is really important for understanding how protons can affect the ionization states of common functional groups. And we've seen how these functional groups are really important for maintaining the structure of these biologically active macromolecules. We can make buffers in the lab using a variety of chemicals, and these are going to help buffer those acid-base reactions from occurring. We can use the Henderson-Hasselbalch equation to determine the precise pH of a mixture of a weak acid and a base found in a buffer, and it's really important to control the ionization state of a protein's functional groups in order to maintain its structure and/or function. So the last thing I want you to think about is that lysosomal enzymes are actually found in these lower pH environments. So what sort of changes could you make to a protein structure in order to help it maintain its function and stability at these low pH values? Don't forget to review the lecture before you go on and complete the brain break questions on Quercus.